Welcome to Exercise 26, Food and Milk Quality Introduction. You should turn in your lab manual to page 235. The CDC, or Center for Disease Control, reports that the top 5 microbes that cause food poisoning are Salmonella, Clostridium, Campylobacter, Staphylococcus aureus, and norovirus. The CDC estimates that each year 48 million people are infected with a foodborne pathogen and become ill. Food can become contaminated when irrigation waters that contain feces, also during butchering process of meat, and the food preparation in restaurants and in factories. So let's go back to the part about the butchering and meat. So if you remember, when we did our experiment of coliforms from our water experiment, we were looking to see if coliforms were present, things like E. coli and Enterobacter. One of the characteristics is they were in warm-blooded animals, and they were gram-pot or gram-negative, and they were lactose fermenters. So what we're seeing here is that it's possible during the butchering of the animal, the intestine of the animal is nicked, and some of those coliforms from the intestines could potentially get into our food. To prevent contamination, food is washed, boiled, pasteurized, and chemical preservatives are added. The elderly, young, and immunocompromised are the most susceptible to foodborne illnesses. Raw milk is unpasteurized milk and could contain microbes. Raw milk could contain coliforms from the udders of cows. Again, the feces of cows is near the udder, and those feces could contain coliforms. So to prevent from getting sick from raw milk, we pasteurize milk. Pasteurized milk is heated to temperatures that kill microbes but doesn't damage the taste. So in exercise 26, the goal is for us to look at food and milk and to see, using the standard plate count, how many colonies of microbes are present. What we will not be able to identify in either one of these experiments is what bacteria is present. We will be using nutrient agar, which pretty much grows any bacteria. So exercise 26, the setup of food and milk experiments. Please turn in your lab manual to page 237 and then 238. So let's talk about the food procedure first. A sample of milk or food is pipetted into a water or dilution bottle containing 99 ml of water. A serum dilution is made. 1 ml and 0.1 ml of the dilutions are pipetted into plates. Nutrient agar is added to the plates and allowed to solidify. So let's view figure 26.3. So in this figure, we see 1 gram of food, which is equivalent to 1 milliliter of food, is being added to a 99 ml dilution bottle. If you recall, our formula is the volume added divided by the total. The volume added in the bottle is 1 gram or 1 milliliter. The total would be 100. The dilution on that first bottle is 10 to the negative 2. We then take out 1 ml, our pipette, into the first plate, and so therefore 1 ml times 10 to the negative 2 is still 10 to the negative 2. The second plate, we're going to withdraw our pipette 0.1 ml, which gives us our dilution of 10 to the negative 3. We repeat the exact same procedure using the milk samples. Some of the groups will have pasteurized milk, and the other half of the groups will have unpasteurized milk. This procedure is exactly the same as the food. The only difference is the samples that are being used. We also want to make sure that we use gloves to not contaminate the milk or the food samples. Exercise 26, Purpose and Hypothesis. So on pages 237 and 238, you need to fill in what you think the purpose and hypotheses of these two experiments. So groups 1 and 3 are going to test canned green beans with an expiration date of May 4th. Groups 2 and 5 will test frozen carrots with an expiration date of 5/20. Groups 4 and 6 will test ground beef with an expiration date of March 16th. Now we're going to assume this experiment is being done in real time. So that would mean the experiment was being done the week of March the 30th. Fill in the purpose and hypothesis in the food experiment on page 237. You may be submitting this on Blackboard for points. So when you go back and look at the types of foods that we're testing, you need to see what some of the variables are. We have canned green beans, which are at room temperature. We have frozen carrots, which are obviously frozen, and then ground beef. So one of those comes from an animal, and think about enzymatic activity that we discussed in lecture and lab when it comes to cold temperatures. And also what we talked about as far as what happens when food is heated or cold or where it comes from. Groups 1, 3, and 5 are going to also test pasteurized milk with an expiration date of April 28th. Groups 2, 4, and 6 are going to test raw or unpasteurized milk with no expiration date. So on pages 237 and 238, please write down what you think is going to happen for this experiment. You're going to write the purpose and hypothesis for the food and the milk experiments on pages 237 and 238 respectively. So think about what you know about food and what we've discussed in lecture. Exercise 26, food data results. So since we're not actually doing this experiment in the lab, I'm going to give you hypothetical data. You're going to use the hypothetical data as if you have the plates in front of you in the laboratory. Using the data below, calculate the OCC, original cell concentration, of each type of food or milk. So what this is telling us is how many bacteria is actually in this case, the food samples. Again, we will not know what kind of microbes are present or bacteria, just that there are microbes present. Using the data below, fill in the table on page 239. So in your lab manual, go to page 239, and you're going to write down the number of colonies for each of the different types of foods. So let's look at our data. Canned beans yielded 63 colonies on our 10 to the negative 2 plate. 5 colonies were seen on the 10 to the negative 3 plate. And that's logical because the more dilute, the less colonies. Frozen carrots, we had 28 colonies on the 10 to the negative 2 plate, 2 colonies on our 10 to the negative 3 plate. And again, you're filling this data in lab and you will be, or in your lab manual, and you will be submitting your answers to the analysis questions on Blackboard. The last set was for ground beef. The first plate, which was 10 to the negative 2, had over 300 colonies. And the 10 to the negative 3 plate had 199 colonies. So at this point, you need to recall, what is our magic number for using the number of colonies? 30 to 300. So which of these plates contained too few to count, too numerous to count, or in that sweet spot of 30 to 300 colonies? Page 239 is where you're going to calculate the results of the food experiment. So at the top is a refresher of how to do the formulas. So the formula that we actually want to use is the original cell concentration. Again, that is going to tell us how many bacteria are present in the beans, carrots, and ground beef. Our formula is the number of colonies, which you see on the plates below, times the dilution factor, or DF. That is going to give us our units, or OCC, which is the number of cells per milliliter. Before you can do your OCC, you need to recall that the dilution factor is 1 over the dilution, or the reciprocal of the dilution. So for example, if our dilution was 10 to the negative 2, then our dilution factor is 10 to the 2. The top goes through some examples in that table. Also recall that you must put all of your answers in scientific notation. Milk data. So on page 239, we're going to do the exact same thing we did with the food, but now we're going to use the data for the milk experiment. Again, half of the groups used unpasteurized or raw milk, and the other half used pasteurized milk. So the difference is the heating up of that milk. Using the data below, calculate the OCC for each type of food, in this case milk. Using the data below, fill in the table on page 239. So the very first plate for the unpasteurized milk had over 300 colonies on our dilution of 10 to the negative 2. That would be too many or too numerous to count. Our second plate with the dilution of 10 to the negative 3 had 298 colonies. So you're going to fill that in on the table. We have pasteurized milk. We have 5 colonies on our 10 to the negative 2 plate and 2 colonies on our 10 to the negative 3 plate. So think about what we know about our quote-unquote magic number and if that will work for our table in calculating the OCC. Exercise 26, calculating milk results, page 239. This is the exact same concept that we did with the food. You're going to take the number of colonies, if it's between 30 and 300, multiply it by the dilution factor. So you take the dilution and do the reciprocal or inverse. Dilution factors are whole numbers or positive exponents. Dilutions are negative exponents. That's going to give you your OCC, or cells per milliliter. Once you've done that, we're going to do the analysis on the next page. Exercise 26, milk, the methylene blue reductase test. You should be in the lab manual on page 236. In this experiment, this is looking at another aspect to see if milk is good quality or poor quality, or pasteurized or unpasteurized. In this test, 1 milliliter of methylene blue solution is added to 9 mils of pasteurized milk in a test tube. Another test tube contains 1 milliliter of methylene blue solution and 9 mils of unpasteurized milk. Your lab instructor will set this up as a demo. Test tubes are then inoculated and incubated at 38 degrees for 48 hours. You will then look at their results and analyze them on the next few slides. Exercise 26, Food and Milk Conclusion and Case Studies. You should be in the lab manual on pages 240-241. Complete conclusion questions 1 through 5 on page 240. Complete case study questions 1 through 8 on page 241. These questions will be submitted on Blackboard. Instructions on how to submit them on Blackboard will be detailed. Note, page 242, the metabolism video is being omitted for this semester. Take the Lab Quiz 6 on mastering using the Blackboard site. This will be over all of the exercises. Exercise 24, dilutions, exercise 25, the food and the milk, and exercise 26, also the E. coli experiment. Hope this helps.